Hello everyone, I welcome all of you to my channel. This is MEENU AGRAWAL. Today's topic is cell membrane. So about cell membrane, you know that this is the very important part of your cell. So suppose if I draw a cell, no? I'm just making for you. Suppose this is your cell membrane, isn't it? Now this is what? This is forming your what? It's a boundary. It's a boundary which separates your cell from external environment. It separates the cell from external environment. Okay? This is something very important, that it gives what? Identity to the cell. Shape to the cell. उसको क्या दे रहा है? Shape दे रहा है. Isn't it? This is something very important, and this is the structure which is maintaining. It decides. This is the membrane which decides what has to move inside the cell and what has to move outside the cell. Okay, now before coming to their function and all, I will discuss about these function also in detail. Therefore, I have divided this plasma membrane into sub videos. Okay, so first of all, let's look at what our membrane is made up of. So it is made up of what? It is made up of your lipids, and it is second thing, it is made up of your proteins. Okay, lipids and proteins. So first I will talk you about these lipid molecules. How do these lipid molecules are? Little bit you people might be knowing about it, that lipids are what? Lipids are your fats. They're nothing but your fats, or you can say oils. You must be seeing so many, and you know about lipids, that they are what? Lipids are what? They are, lipids are, you can even do this experiment and can see it on your own also. Don't believe me. Just if you pour any oil on water, okay? So if you pour that oil on water, it is what? It floats. It floats on water. Means it is what? It is what? Lighter. It is lighter than water. Therefore, it is floating on water, isn't it? And if you look at this molecule, I will little bit talk about its chemistry, because to understand the structure, you have to understand its structure, a little bit about the chemistry of this molecule. Now, if I draw this molecule, it has got a head, okay, and it has got a tail. Okay, it has got 3 tails. Okay, there are different kind of lipid molecules. When we will go to biomolecules chapter, we will read about them. But just simple basic idea I am giving you about here, that this, if you look at this lipid molecule, if you look at this lipid molecule, what it has? It has got a head. This head is what? What is the property of this head? It is what? It is, you must have heard about hydro. Hydro मतलब सब कोई समझते हैं मेरे ख्याल से. hydro मतलब water. Hydro means what? Water. And it is what? Hydrophilic. Philic means loving. So it is water loving. And if you look at this region, which is your tail, this is your hydrophobic, है ना? कई लोगों को फोबिया होता है, डर लगता है कई चीजों से, that is phobic. So they are what? They're water repelling, है ना? वो क्या करते हैं? वो water से दूर भागते हैं, because they don't like water, है ना? And in chemistry, see, in life also we have a chemistry, isn't it? कई लोग हमें पसंद होते हैं, कई लोग हमें नहीं पसंद होते। जो पसंद होते हैं हम उनके साथ जाना चाहते हैं, और जो नहीं पसंद होते उनके पास तो हम बैठना भी नहीं चाहते। So something with this molecule is also like that. Okay, now I'm raising this question from all of you, like um, so let's see ki, suppose this is a beaker or any glass which is having water. Okay, it is having water. Now if you pour oil on it, okay, if you pour oil on it, then what will happen? All of you know that it will form a layer, it will form a film of your lipid on top of this water, isn't it? Now if I ask you how will you show that these molecules are on water, then what will you do? Huh? If this is your water, and then what will I, how will these molecules arrange? Think. Your head will face towards water. इसका जो head है वो पानी की तरफ होगा, क्योंकि वो हाइड्रोफिलिक है ना। और जो टेल है वो हाइड्रोफोबिक है, वो उसको लाइक नहीं करता है, इसलिए वो उससे दूर जाएगा, है ना। तो जो बच्चा आपको पसंद नहीं होता उससे आप दूर बैठते हैं, ठीक। दिस इज द केस हियर। नाउ अगर इस ऑयल को मैं मिक्स कर दूं ग्लास रॉड से, या किसी चम्मच किसी से भी घर में आप करके देखिए, जितना आप करेंगे साइंस में उतना आप सीखेंगे। तो इट्स वेरी इंपॉर्टेंट दैट यू डू इट। और इसको आप पानी को में अगर मिक्स कर देंगे तो आपको क्या मिलेगा? आपको कुछ इस तरह से मिलेगा कि ये जो फैट है ये ड्रॉपलेट्स के फॉर्म में आपको पानी के बीच में दिखेंगे। तब अब ये मॉलिक्यूल्स कैसे अरेंज होंगे, सोचिए। हाँ, कैसे अरेंज होंगे? हाउ विल दीज मॉलिक्यूल्स विल बी नाउ अरेंजिंग देमसेल्व्स इन वाटर। ओके, तो ये अपने आप को पानी में कैसे अरेंज करेंगे अब? तो इसको जरा से मैं आपको दिखाती हूँ बना के। आप देख सकते हैं आपका हेड बाहर होगा, ठीक। तो ये एक ग्लोब्यूल के फॉर्म में है, आप देख रहे हैं ड्रॉपलेट के फॉर्म में है। तो आप क्या देख रहे हैं कि इस तरह से ये गोल सा हेड बना लेगा। ओके, just make me larger, I'm just making this one a larger outside. ओके, सो ये आपको हेड बना लेगा, ठीक, इस तरह से ड्रॉपलेट के फॉर्म में। और ये अंदर में टेल बन जाएंगी इसकी। तो जितनी भी टेल हैं ये सब इधर आ जाएंगी। सो मेकिंग अ बिगर वन फॉर यू, अनदर वन। ओके, इस तरह से आप बना लीजिए। ओके, और ये हो गया टेल, ठीक है। तो ये आपने केमिस्ट्री में भी अगर ये स्ट्रक्चर पढ़ा है तो वी कॉल इट मिसली, isn't it? तो जितने भी हेड्स हैं वो सब बाहर की तरफ आ गए, जहां पे कि क्या है, एवरीवेयर इज वाटर देयर आउटसाइड। तो बाहर पानी है तो दे आर फेसिंग एंड द एंड्स आर फेसिंग टुवर्ड्स इनसाइड। नाउ आई एम मूविंग टुवर्ड्स द स्ट्रक्चर ऑफ योर membrane. Okay, now if we have the structure of membrane. Now just I am showing you here pictorially, you can see here, and see this is in 3 dimension. And remember जब भी हम सेल की बात करते हैं तो it's in a 3 dimension. To remember it's like a balloon, जैसा बलून होता है ना वैसा स्ट्रक्चर होता है। It's a 3 dimensional, it's not a 2 dimensional structure. ओके, सो यहाँ पे आप देख सकते हैं कि कैसे ये मिसली 3 डायमेंशनल स्ट्रक्चर के फॉर्म में आपको दिख रहा है ना। और ये जो मेम्ब्रेन है, अब मेम्ब्रेन के साथ क्या प्रॉब्लम आती है? इफ यू लुक एट अ मेम्ब्रेन, व्हाट डू यू फाइंड? in the membrane there is an outside water is outside also there, and water is inside also there. Then how will these molecules arrange themselves? अगर ये हेड इधर है तो वाटर है, और टेल इधर है तो भी वाटर है, then it's a problem. Then to solve this problem, now it was your Nicholson and Sanger, they proposed the fluid mosaic model, and they said that the lipid molecules is in the form of a bilayer. वो किस फॉर्म में है? बायलेयर पे है। ये देख रहे हैं ना, एक लेयर है और ये नीचे में एक और लेयर है ना। So if I have to draw it, how will I draw it? जस्ट शोइंग यू, आपने एक लेयर बनाया और ये दूसरी लेयर बनाया. ओके, both are heads and these are your tails. Okay, these are your tails if you look at them closely. Okay, ये इस तरह से आपको बन रहे हैं. Okay, so outside is also water. Okay, and inside it's is also water. Your cytoplasm is also having a high content of water, and also your extracellular fluid is also having high content of water. You can think like this, na? Structure is there, and you can see that these compartments, if you look at in your cell, like your organelles are also there, so they form structures like the membrane forms structures like this. Okay, now moving on to the structure of the cell membrane. Just I am trying to show you. Yes, now you can see here. Okay, this is your bilipid layer. Okay, और आपको दिख रहा है ना, it has got a head and it has got a tail. Okay, now this molecule, the kind of lipid that is present in your membrane is what? It's called as what? It is your phospholipid. ये कौन सा तरह का लिपिड है? It's a phospholipid. Okay, it is a phospholipid. Okay, now majority of them are your phospholipid. Now if you look at these now lipid molecules, you are right now not knowing about saturated and unsaturated, but remember that these lipids provide fluidity. What? Fluidity. जैसे आप देखते हैं ना, ऑयल कैसा होता है? फ्लूइड होता है ना? तो it's fluid like structure. So this membrane is also a fluid like structure, because I just will tell you about an experiment which was done, and it was seen that it is a fluid like structure. Okay, I will come to that experiment also. Now just look at the components of the membrane first, and what these are your membrane, and these are what? These are your cholesterol. As you can see here, these are your cholesterol. You know that cholesterol is also a kind of a lipid, and it is see, this cholesterol and the tail of the phospholipid, it all they are very important in determining the structure of your membrane. Okay, the more the longer the lipid length is, it makes the membrane less flexible. Okay, and the shorter the length is, the membrane becomes more flexible. Okay, and you will see that if there is more unsaturated fatty acid, it makes it what? More flexible. Okay, because now it has been seen, and remember that the length of your membrane is how much? On an average it is 7 nanometer, and this as the temperature decreases, अगर temperature जैसे colder regions पे जो organisms रह रहे हैं, वहाँ पे temperature जब कम हो जाता है, तो ये क्या होगा? ये oil जो है saturated अगर ज़्यादा है fatty acids उसमें, तो वो जम जाएगा आपको. ठीक है, तो रिमेम्बर जैसे घी हुई होता है ना, जाड़ों में जम जाता है, आप देखते हैं ना. तो यहाँ पे इसको सुलझाने के लिए ये प्रॉब्लम क्या हुआ है? देयर मेम्ब्रेन इज मेड अप ऑफ मोर ऑफ अनसैचुरेटेड फैटी एसिड. आप ऐसे समझ सकते हैं सिंपल भाषा में, जैसे जो ऑयल हम लेते हैं, है ना, प्लांट सोर्स से जो ऑयल मिलता है, जैसे सोयाबीन है, ये आप देखते हैं जाने में भी पिघला हुआ रहता है ना. तो समझ जाइए कुछ इसी टाइप का मॉलिक्यूल सैचुरेटेड अनसैचुरेटेड फैटी एसिड्स का ये फॉस्फोलिपिड बना होता है, इसलिए वो कोल्डर रीजन्स में भी वहां पे उनके क्या होता है, वो जमता नहीं है. तो दिस इज एन अल्टरनेटिव द नेचर हैज टेकन अ सब्स्टिट्यूट फॉर इट. और जैसे कि आप देखेंगे कोलेस्ट्रॉल आपको ज्यादातर एनिमल्स में मिलेगा, प्लांट में, बैक्टीरिया में तो एकदम ही नहीं मिलेगा आपको. तो ये जो कोलेस्ट्रॉल है, जो कि बहुत ही जब गर्म हो जाता है, तो वहां पे ये कोलेस्ट्रॉल क्या करता है, वो एक तरह से उसको जमाने में मदद करता है, उस मेम्ब्रेन को कुछ ज्यादा ही फ्लेक्सिबल हो जाएगा फिर वो, है ना. तो उसको एक मेंटेन करता है, उसकी वो क्या कंसिस्टेंसी को मेम्ब्रेन के. ओके, उसकी फ्लूइडिटी को मेंटेन करता है. ओके, सो दिस इज व्हाट वी हैव सीन. ओके, तो वो उसी प्रोपोर्शन में होता है, सो दैट कि ना membrane structure is maintained. Okay, so it gives mechanical stability, rather you can use the word that it cholesterol is helping in giving what? Mechanical stability. Huh? To the membrane. Okay, so और ये क्या होता है, और quickly burst नहीं होने देता है cells को, ना, it prevents, ना, this cholesterol I'm talking about, it prevents the cell from bursting. It prevents the cell from bursting. It prevents cells from bursting. Okay, so ये सारी चीजें होती हैं, ठीक है। तो हर एक जो कंपोनेंट है ना, वो उसका बहुत ही इम्पोर्टेंट पार्ट है। हाँ, और ये जो हाइड्रोफोबिक रीजन है इसका, ये जो पार्ट है इसका, आप देख रहे हैं कोलेस्ट्रॉल का ये जो तेल है, ये हाइड्रोफोबिक रीजन है। ये क्या करता है, ये प्रिवेंट करता है ना ionic molecules or polar molecules. You will just polar molecules means you can understand that they are attracted towards water. Okay, so you can say that they prevent ions and water soluble rather substances to pass through. तो उन substances को they prevent, they don't allow that to pass through. Even your lipid layer, na, it is preventing, na, water soluble substances to pass through the membrane. Okay, they will allow lipid soluble चीजें आराम से pass हो जाएंगी, पर water soluble substances no, they won't be able to pass through the membrane very easily. Now what else other things your membrane is having, let's look at that. Okay, so now you can see here that these are the these are what, these are what, these are the proteins which are present in your membrane. Okay, and these proteins are what, they are of two types. What are they? They are basically your integral protein, or intrinsic protein we call them, and the other one is called as your extrinsic protein. Okay, so they are your integral proteins, okay, and they are just embedded in your membrane. Okay, they are not loosely attached to your membrane. Okay, so they are tightly attached to your membrane, and they can just like I can say you like an iceberg में आप देखते हैं ना, सी में icebergs होते हैं, आइस के छोटे छोटे टुकड़े होते हैं, वो फ्लोट करते रहते हैं, है ना, तो दे ऑल्सो मूव अलोंग द मेम्ब्रेन। तो उसी तरह ये भी मूव करते रहते हैं आपके मेम्ब्रेन में, ठीक है। तो दीज आर इंटीग्रल अलोंग विद दैट लिपिड ना, ये भी मूव करते रहेंगे। तो ये इंटीग्रल प्रोटीन्स हैं, okay, and they are present on both the ends, as you can see here. They are present on inner part also and the outside part also, and they are also transmembrane protein. Okay, they are your transmembrane protein. Even they form a transport protein. Okay, now your extrinsic proteins, they are loosely attached, okay, to your membrane. Okay, they are even towards the water molecules that is present towards inside or outside. They interact with them also, and they can be even taken away. Okay, so there are two kind of proteins that I have mentioned. One is your integral protein and the other is your peripheral proteins. And these proteins play very important role when see these proteins acts as enzymes, as channels. They act as like water soluble substances can't pass through this lipid layer, then how will they pass? They pass through these proteins. Okay, so these proteins are very important, so we will talk about them later. In the later video I will talk about them a lot. Now this is about your what, these are your what, these are your, you can see as the structure is telling you, they are your sugar moieties. Since they are attached to a protein, we call them as what, we call them as glycoprotein. ये क्या हो गया, ये हो गया glycoprotein. और चूंकि ये लिपिड से अटैच्ड है, आप देख सकते हैं यहाँ पे, देखिए ये लिपिड से अटैच्ड है ग्लूकोज, तो इसलिए इसको क्या बोल रहा है, इसको ग्लाइकोलिपिड बोल रहे हैं. Okay, so like that they are being called. Okay, now but these now molecules, they are having very important role. Okay, like I was just saying about, let me move, that these lipids na molecules, they are playing very important role. Okay, now if you look at them, these glycoproteins, if you look at, or glycolipids, if you look at, they play very important role in cell recognition. Okay, and cell recognition and cell adhesion. Now if you think कि how your ना cells like muscle अपनी जगह पे कैसे है, कैसे वो स्किन से सेल के साथ जुड़ा हुआ है ना, कैसे हो रहा है ये चीज. तो an experiment was done where what they did है ना, उन्होंने they meshed the poriferans, एक बार नहीं कई बार कर दिया उनको, है ना. poriferans तो आप जानते ही हैं, they are also multicellular and they have got very few डिफरेंट काइंड ऑफ टिश्यूज ऑन देम. तो उसको क्या किया उन्होंने, सिंपल बॉडी होता है तो उसको उन्होंने मैश कर दिया. मैश करने के बाद उन्होंने जब वापस से ना उनको सेल्स को छोड़ दिया उसी उसी सोल्यूशन में, तो उन्होंने क्या देखा, थोड़ी देर से वापस से वो फिर से पॉडीफेरा बन गया, फिर से वो बन गया. तो हाउ इज इट दैट कि वो, और फिर एक और एक्सपेरिमेंट किया उन्होंने. एक एक्सपेरिमेंट ये किया कि उसको पॉरीफेरा को लिया, उसको मैश किया, हाँ, लेफ्ट फॉर सम टाइम, and they got the back the porifera, they got back porifera. Okay, and next experiment उन्होंने क्या किया, the next experiment that they did was that what they did, they took two different species of porifera. Okay, दो species लिया porifera का. Suppose I just draw for you any poriferan, just for your shake I'm just drawing it. Okay, so suppose any poriferan if you take, okay, I'm just drawing it. Okay, now this is there. Now this is one, and again another one I'm taking. Okay, now this is your blue color I'm showing you. Okay, now if these two poriferans are there, okay, now they are being meshed, so you get both the kind of cells there. Okay, now और उन्होंने क्या किया, इसको पहले आधा कर दिया, हाफ, कट इट हाफ, और इसके हाफ पार्ट को लिया मैश करके और इसके हाफ पार्ट को लिया. Now what they got the half part of this porifera together, and they got the half part of this porifera together, half part of this porifera together. So how is it that they are quite species specific also, है ना. They are cell specific as well as they are tissue specific, and they are even species specific. ओके, तो वो अपने सेम टाइप को रिकॉग्नाइज कर पा रहे थे और वो एक ही जैसे वाले से जुड़ रहे थे. तो दे प्ले वेरी इंपॉर्टेंट रोल इन टिश्यू रिकॉग्निशन, हाँ, एंड इन ना जस्ट दे आल्सो अ लॉट ना दे सेल सिग्नलिंग में काम करते हैं. वी विल टॉक अबाउट सेल सिग्नलिंग. तो ये दे प्ले अ वेरी इंपॉर्टेंट रोल इन दिस. So this was the question which I was raising in front of you. Now one more thing I will tell you before I move to this. Like I told you about the fluidity of the membrane, so I said that I will tell you. Then an experiment was done earlier. They took a mouse cell and a human cell. Okay, now what they did, they tagged the mouse protein with green dye. They tagged the, उन्होंने उसको ग्रीन डाई से कर दिया. ओके, टैग कर दिया ग्रीन डाई, देखने में वो ग्रीन दिख रहा था ना माइक्रोस्कोप के अंदर. एंड दे टैग्ड द ह्यूमन, दिस इज योर माउस cell, and this is your human cell. Okay, now what they did, they tagged the human cell with the red dye. Red dye से कर दिया, huh? And then they fused these two cells. They fused the two cells, and what they got for them, this is the, so this is your green dyes available here, and this is your red dye. And after some time, they left it for half an hour, and to their surprise, what they got? What they got? They got that red and blue dyes have mixed together, which shows what? That the membrane is highly fluid. अगर वो fluid structure नहीं होता तो क्या ये मिलता? और इसका बहुत बड़ा उदाहरण है. See, look, if our membrane would have been a rigid structure, क्या we would have been able to move our body? No. This shows that the membrane is highly flexible structure. Okay. So let's now talk about the various things that we have discussed today. In a very brief, I will just summarize it, okay, for you people. Just see these biological molecules, which consist of what? It consists of your as you can see here, it consists of your lipids, proteins and carbohydrates. Okay. The fluid mosaic model, it was proposed by Nicholas and Sanger, describes that phospholipid bilayer in which proteins can move about within the plane of the membrane. Okay. Now, this is the second slide of mine, which tells you about that the two leaflets of the membrane. Two leaflets of the membrane means the upper side of the membrane. I told you, no, it's like this. So if I take this side and the another side of the membrane, okay, these two different sides, now they are what? They are different, है ना. It's not that कि ऊपर में जो प्रोटीन है वही नीचे वाले भी तरफ में होगा. No. Okay, so they are quite different, and the next is your carbohydrates attached to the proteins. As I just now told you, they are what? Glycoproteins if they are attached to proteins, and they are glycolipids if they are attached to your lipid molecule, and form the external surface of your plasma membrane and functions as recognition signals, and they help in assembling your cells. ना cells को recognize करने में recognition signals भेजता है ना. and like अगर आपका ही cell cancerous हो जाता है तो आपके जो आपका इम्यून सिस्टम है, आपका जो WBC है, उसको रिकॉग्नाइज करने लगता है कि यहाँ पे कुछ ये फॉरेन मटेरियल सा आ गया है. ओके, सो इन ऑर्डर फॉर सेल्स टू असेंबल इनटू टिश्यूज दे मस्ट रिकॉग्नाइज एंड एडहर टू वन अदर, है ना. तो ये बहुत ही इंपॉर्टेंट फैक्टर है कि वो एक दूसरे को से जुड़े, है ना. नहीं तो एक सेल दूसरे सेल से ये पूरा टिश्यूज का फॉर्मेशन ही कैसे होगा अगर वो नहीं जुड़ेंगे तो, है ना. और पूरा ऑर्गन का फॉर्मेशन कैसे होगा, ये सब इस पे ही डिपेंड करता है. एंड लास्ट नॉट द लीस्ट इज योर व्हाट इज दैट membrane structures are not static structures. They are constantly forming, exchanging and breaking. Exchanging क्यों? क्योंकि कुछ आ रहा है सेल के अंदर, कुछ बाहर जा रहा है सेल से. और ब्रेकिंग डाउन भी है क्योंकि कुछ जब निकल रहा है तो वो वहां से वो पार्ट ब्रेक कर रहा है. तो on we will talk about it in detail because we will deal with active and passive transport as well. So there are various dimensions of your membrane. Remember that membrane is a highly living, changing, dynamic structure. It keeps on changing, okay, and so many things it does. Okay, so with this introductory part of the structure of the membrane I'm ending. And so don't forget to subscribe. Goodbye.